My project aims to find the role of CHD6 in the oxidative stress response. DNA is organized in a structure called chromatin. The structure of chromatin is depicted in the figure to the left. Chromatin is composed of DNA wrapped around a histone protein octamer core, or a nucleosome. As seen in the figure, 8 proteins form a ball with protrusions sticking into the DNA. Nucleosomes secure the DNA, allowing it to be stored compactly. However, the nucleosome structure prevents effective repair of the DNA, as it cannot be accessed, so it must be altered for repair to occur. Failure to repair damaged chromatin increases genome instability and drives cancer formation. Chromatin comes in 2 varieties, euchromatin and heterochromatin. Euchromatin can be read by the cell and is important in the transcription of proteins and cell function. Heterochromatin is very compact and, while not expressed, plays a major role in cell division. The second figure to the left depicts a DAPI stain of 2 nucleases. The brightness of the dye correlates to the amount of DNA present. The very bright areas show where heterochromatin is located, as this area contains lots of condensed DNA. Damage to the structure of chromatin can result in genetic instability, which can lead to the cell becoming cancerous. CHD6 is a chromatin remodeling enzyme known to be involved in DNA repair and cancer driver genes. CHD6 works to move nucleosomes along the DNA or remove them entirely. This sliding activity of CHD6 exposes DNA, allowing it to be transcribed or repaired depending on cellular context. This process is depicted by this figure, where histones can be inserted, evicted, exchanged, or slid along the DNA. CHD6 also plays a role in the transcription of antioxidant genes, which are important in cell survival and typically misregulated in cancers. However, the specifics on how CHD6 interacts with other proteins and repairs DNA is unknown. The study of CHD6 is vital to the understanding of the formations of many cancers, as curiously, CHD6 is amplified and mutated in numerous cancers, at least while compared to wild-type tissue. Specifically, skin and bladder cancers are subject to many mutations and amplifications respectively. The biological effects of this amplification are poorly understood. However, it is known that CHD6 does play a role in the transcription of antioxidant proteins. So mutation in the CHD6 gene could potentially disrupt the balance of radicals and antioxidants in the body. Knowing the protein-protein interactors of CHD6 is vitally important in the study of cancer and cancer formation. Chromatin remodeling enzymes need interacting partners to direct them to certain DNA sequences, as well as interacting partners that control their enzymatic activity either through direct interactions or post-translational modifications. Validating these potential interactors would expand our knowledge on the function of CHD6 and provide insight into its role in the formations of these cancers and DNA repair pathways. Because of CHD6's vital role in the study of cancer and genetic instability, my research question is, what are the protein-protein interactors of CHD6? This experiment will determine the protein interactors of CHD6 with the use of CRISPR knock-in and proximity ligation assays. A proximity ligation assay, or PLA for short, causes proteins to fluoresce when in contact with an interactor being tested by using antibodies. In this experiment, 6 potential interactors will be tested. These interactors are shown in the figure in red. PRMT1, HMGB1, SUPT16H, CBX3, and PARP1 are all chromatin remodeling enzymes, with very well characterized functions. PARP1 and HMGB1 have characterized roles in DNA repair. PRDX1 is an important redox sensor in the cell. The proteins shown in blue are non-specific interactors and will not be tested in this study. To set up a PLA, a CRISPR system must first be used to knock in a tag on CHD6. This will allow us to monitor its presence in the cell. In this case, a CRISPR system must first be used, as there are no good antibodies that work against endogenous CHD6 that are commercially available. The CRISPR knock-in works by creating a double-strand break in the DNA sequence for CHD6. This is done with the use of a protein called SPRY. The cells work to repair the damage, but homology directed at repair occurs with a donor sequence that we will add during the CRISPR process. The donor sequence will include a flag tag as well as the homology for the CHD6 gene. During repair, the cell will use a donor sequence as a template for repair and insert the flag tag while doing so. SSODN will be the donor sequence used for CRISPR. It is shown in pink on the diagram for the sequence. The 3 different guides that will be used to carry out CRISPR are also shown. The corresponding sequence for the 3 gRNAs are above their respective bars. Guide 1 is in yellow, Guide 2 is in blue, and Guide 3 is in green. Guides 1 and 2 will be disrupted by the insertion of the flag tag, so there's no possibility to recut following the insertion, which will ensure accurate results. A lentipuroplasmid is a plasmid that provides a cell with resistance against the antibiotic puromycin, and it also contains a DNA sequence for the production of lentiviruses. The gRNAs from SSODN were inserted into a lentiguide puroplasmid. This plasmid was then inserted into an HEK293 T cell with the process of lipofection, wherein the gRNA sequence is surrounded by lipids so it can diffuse across the plasma membrane of a cell. After 48 hours, the lentivirus was harvested and used to infect RPE1 cells. RPE1 cells will be used for the proximity ligation assay. Since a lentipuroplasmid provides puromycin resistance to RPE1 cells, using a kill curve, it is possible to determine the concentration of puromycin that will kill non-infected cells. At this dose, it is possible to select for infected cells that will be used to carry out PLAs. The optimal dose was determined to be from 4 to 6 micrograms per milliliter range of puromycin. And we opted to use a 6 microgram per milliliter dose, as there was more cell death at this higher dose. On this graph, it is hard to see, as the scale of the graph drops dramatically after puromycin was added. As of now, this is what has been accomplished so far. Going forwards, SPRY, an enzyme related to the traditional Cas9 usually used for CRISPR, will be added alongside a single-stranded DNA donor containing the sequence that will code for the flag tag. This will be administered with the process of electroporation. Successful insertion will be determined using PCR. Antibodies specific to the flag tag will be used to label CHD6, as well as antibodies for their interactors. PLAs will then be used to determine the interactors of CHD6 identified in the screen. These interactors will help determine CHD6's role in the cell. PLAs work using antibodies that are specific to a protein and its interactor. A. 2 primary antibodies recognize the 2 proteins of interest and bind to them. B. Secondary antibodies with oligonucleotide tails bond with the primary antibody. C. If the proteins are within less than 40 nanometers away from each other, connector oligos ligate with the tails, forming closed, circular DNA. D. The resulting closed circular DNA template becomes amplified by the DNA polymerase. This will cause close to 1,000-fold signal amplification and signal detection even in low-abundant proteins such as CHD6. In step E, detection oligos with fluorochromos hybridize to repeating sequences in the amplifications. Finally, the interaction can be detected using fluorescence microscopy. In the representative figure, DAPI, a DNA binding dye, is used to visualize the nucleus. Separate antibodies are used to visualize proteins in the cytoplasm, as seen in green. The red signal represents successful rolling cycle amplification for the PLAs. And these red dots, or foci, can be counted and compared to negative controls and determine whether we are observing true protein-protein interaction. The controls we will use for a PLA are called technical controls, and they will assess whether we obtain any rolling circle amplification from the individual antibodies alone. They are the following: anti-flag alone for CHD6, antibody against purative interactor alone, and no primary antibodies. This will assess the background amplification that occurs. However, it is typically 0. I would like to give a quick thank you to Dr. Erin Godarzi for providing the materials and lab space required for this experiment, and Luc Povinchet for his guidance and step-by-step instruction, as well as Dr. Garcia for her support and guidance. Questions?